Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we're going to be talking about the bacterial growth and life cycle. So bacteria reproduce primarily through binary fission. This is an asexual process in which a single cell divides into two identical daughter cells. Before division, the bacterial chromosome replicates, and each copy is attached to a different part of the cell membrane. As the cell elongates, the chromosomes are pulled apart and the membrane pinches inward to form two separate cells. This simple and efficient mode of reproduction allows for rapid population growth. However, the rate of reproduction depends on environmental conditions, and this is reflected in the bacterial growth curve. This describes population changes over time in a closed system, which would be a batch culture. Under ideal conditions, population growth is exponential. So let's talk about the phases of a bacterial growth curve. So the first phase is a lag phase. This is prior to exponential growth, and here bacteria are metabolically active, but they are not dividing as they adjust to the new environment. After this, you have the log phase, or the exponential phase. Here, cells divide at a constant and rapid rate, and this is when bacteria are most metabolically active and most susceptible to antibiotics. After this, you have the stationary phase. Here, cells cease to divide because of nutrient depletion and waste accumulation. This causes the rate of cell division to equal the rate of cell death. And finally, you have the death phase. Conditions worsen and the death rate surpasses the division rate. This leads to a decline in population. The maximum population at the stationary phase is called the carrying capacity for that environment. For some bacteria, especially gram-positive genera like Bacillus and Clostridium, the life cycle includes the ability to form endospores. Endospores are highly resistant, dormant structures that form when conditions become unfavorable. Endospores have a thick peptidoglycan layer and can survive extreme heat, desiccation, radiation, and chemical disinfectants. They can survive in temperatures above 100 degrees Celsius, which is why autoclaves or pressure cookers are actually required to completely sterilize liquids and substances that cannot be heated sufficiently in a dry oven. The metabolic reaction of an endospore is called germination, and a single bacterium can only form one spore per cell. When favorable conditions return, the endospore germinates back into a vegetative bacterial cell. This ability gives certain pathogens the resilience to persist in harsh environments. An example would be Clostridium difficile, which is a bacterial infection in hospitals. Overall, bacteria grow by binary fission, progressing through a predictable growth curve in culture. Under stress, some can form durable endospores to survive, and understanding these processes is crucial for microbiology, medicine, and biotechnology. That's it for today. Next time we're going to talk about genetic exchange in bacteria.